Hey, I'm Mr. Williamson. This video is about mitosis and a few other things. Before we get started, make sure you're taking good notes. Again, you're going to need to attach them to the assignment in Google Classroom for credit. Videos now are worth 8 points, 3 points for watching it, and 5 points for having the notes attached. Make sure you're taking good notes. Again, I really like Cornell or outline style, but you do whatever you want to do. Whatever makes your cat meow, do that. Let's get started. Real quick review about the cell life cycle. Again, remember, the cell life cycle is the life cycle of the cell from some point in the cycle to that point back again. In the past, in the previous video, we have talked about this phase over here, the G1 phase, where the cell just basically goes through its cell thing. We talked about the S phase, the synthesis phase, where the cell replicates the DNA, copies, unzips the DNA, T's with G's, C's with C's, etc., things like that. And then finally, we talked about the G2 phase, where the cell prepares for mitosis. What we're going to look at right now is the mitotic phase, the 4 phases of mitosis. All right, it's the actual where the DNA is actually separated. All right, and in there we can throw cytokinesis, which is the splitting of the cell. All right. So before we go any farther, in the previous video I talked about what could, that the cell cycle could go wrong. So what could go wrong in the cell cycle? Well, there are inhibitors and promoters. There are certain enzymes, certain processes that stop the cell from dividing or cause the cell to divide. And if a mutation takes place in the DNA, those inhibitors or promoters don't work. They either don't start something or they don't stop something. All right. Their end result is that cells grow uncontrollably. That's cancer. They ignore a stop signal. Like normally your skin or your cells divide, they stop at a certain point and they become whatever kind of cell they're going to become. But cancer cells don't stop dividing. They keep dividing and they keep dividing. They ignore signals to die. There's actually signals that your body sends out or the cells send out to cause the cell to die when it's time or it's not working right. If it's not doing what it's supposed to do. Well, if that part of the DNA is damaged, then that signal is ignored or it doesn't get sent out. They grow faster than normal. A normal cell grows at a certain rate and reproduces every so many days or even maybe months. Cancer doesn't do that. It reproduces constantly. So it continues to continue to grow. All right. Normally, we would see a cell, something like this, where we start out with the first cell up here, all right, and it divides into 2, mitosis, and then maybe one of those divides, and then something over here, and it goes wrong. Something goes wrong over here, and that becomes a cell that's no good. The signal is sent out, and it dies. That's what the lysosomes do. It causes the cell to die. In a cancer cell, what we have is the cell that's gone wrong, the DNA is wrong, the signals are not sent out, and every time it divides up quickly, and it divides up continuously. So in other words, instead of becoming like 1 cell or 2 cells like here, it becomes 2 cells and 4 cells and 8 cells, and they just keep going. Cancer, though, requires a blood supply. You got to have the glucose. You got to have the oxygen. The body doesn't know what kind of cell it is, just knows it needs to put blood there. So that builds up a whole supply of blood, the capillaries and things like that, and takes resources from other parts of the body. Again, I showed you this in the last video. This is a prostate cancer, an actual photograph of one. These are skin cancers, basal cell, squamous cell, and melanoma. Those are all start out the same way, and the reason that it changes is because of the ultraviolet light that comes down and damages the DNA. It's not repaired correctly. So here's my PSA, public service announcement: wear sunscreen. And if you don't, you end up looking like me. Okay. Another example. This is an X-ray of someone who has osteosarcoma. Osteosarcoma is cancer of the bone, and you can see this is someone's upper, probably their if it's situated right, their left arm, upper arm, and you can see where the bone is not solid. It's it's in that area, the bone is weakened by the tumor, and therefore the bone will not be able to do what it's supposed to do, which is lift your arm and things. It probably breaks real easily. There's a side note. There was in 1951, an African American woman was being treated for cervical cancer. Her name was Henrietta Lacks. She was born in 1920 and she died in 1951 after the diagnosis. She was 31 years old. Her doctor biopsied the tumor without consent. They just, they had consent. They're doing they're trying to treat the cancer. But what he didn't consent was the culture of the cells. He took those cells and he cultured them in the laboratory. And her cells reproduced at a very, very high rate, and they stayed alive. That was the big key. They stayed alive in the laboratory. And they stayed alive long enough to perform different tests and from the same sample. That line of HeLa cells, Henrietta Lacks, that's where the HeLa comes from, was maintained since they were first collected in 1970. Peter Salk, the person who cured polio, had used her cells in the development of the polio vaccine, and they are still used today for research and education. The problem was no one ever told her family that they were using her cells for research. And it wasn't until her family got all kinds of phone calls and requests for blood samples and tissue samples that they were started kind of going, what is this really weird? Why come people are contacting us? And they finally found out that her cells were still being used by research without her or her family's permission. And since then, it has sparked a huge debate over the rights of a patient, to the point where the Supreme Court has determined that if you have tissue that is removed and is considered waste tissue, for instance, cancer or some other tissue that is removed, it no longer belongs to you, and it can be commercialized. It can be used to cure. If in that sample of tissue, if you have the DNA to cure something, it no longer belongs to you. It belongs to the people that they took it. So that's one of the forms you actually sign when you go to the doctor or a hospital, is the right to commercialize your tissues if they choose to do that. Trust me, most of them aren't. But just an interesting side note. Okay, let's talk about review chromosomes real quickly. Okay, normally DNA starts at and spends most of its time as an unraveled, non-condensed state, and it's unraveled state. All right, but like we talked about in the last video, when we start coiling it around these histones, like spools of thread, we can store it on the protein. We take those and we can coil those up until we end up with the chromosome. All right, and you can see here's a different different points of view. Under a light microscope, if we had a really good microscope, you would see something like this here. I'll do that another color for you, something like that. Or under the scanning electron microscope, they look kind of like what's over here. Okay, those are actual photographs of chromosomes, and you can see the fuzziness in these. The fuzziness is actually the DNA that it does in those chromosomes. So remember, when you see a chromosome like this, what you're looking at is replicated DNA. You have the DNA that is on one side is copied over on the DNA on the other side, and what we're trying to do is get this into one cell and this one into another cell. That's the purpose of doing this. All right, here's an image again from electron microscope where you can actually see the shape of the chromosome, but you can also see it because it's kind of unwound. You can see the chromosome shape. You can also see the DNA. This individual strands here are the DNA coiled up. Right? This is a karyotype, like I talked about before, taking all of the chromosomes from one cell. And to do that, most of the time they're done from a woman who is pregnant, and they do amniocentesis. They take a really long needle, from what I understand. I've never been pregnant. Don't plan on doing that. Take a really long needle and they put it into her uterus from the outside, into the belly. and they withdraw some of the amniotic fluid. In that amniotic fluid are some of the cells from the baby, because the baby's cells are there. And they will take some of those cells and they will create this karyotype. All right, and this is a different karyotype that I showed you. The coloring is artificial. The shape is probably very real. All right. Again, each one of these, I said this is something you need to think about. Each one of these pairs, you got one from mom and you got one from dad. All right. You're looking at pairs of chromosomes, one from mom, one from dad. And we're going to talk about that a little bit later. All right. About the chromosomes. When we look at them, we're looking at the homologous chromosomes. Again, the homologous means that one from mom, one from dad. They're the same chromosome, one from each parent. And you can see in here the sister chromatids. The sister chromatids. This chromatid here, I should do that in a little different color. This chromatid here is the exact same DNA as the chromatid next to it. We're going to separate those. All right. Here's the parts of the chromosome that you need to know. I said in the last video you're going to need to know parts of the chromosome. These are the parts. You need to know the sister chromatids, all right, which is the two parts of the X. And you need to know the centromere. The centromere is the middle part right here. So if I drew this, I usually draw chromosomes like an X, okay? The centromere is this middle part right there. The sister chromatids. This is the sister chromatids. Those are the sister chromatids. This is the centromere. All right. So the middle part where they cross is the centromere. The two legs, or the two parts of it, each half, is the sister chromatid. All right. So let's talk about mitosis. Let's talk about it. All right, finally get to what the whole video is supposed to be about. And what mitosis is the answer to what you say when your little sister drops a bowling ball on your foot and she says, "Where did that land?" And you go, "Mitosis." They don't get any better than that. I wait all year to tell that joke. That's the best I got today. Sorry. All right, so mitosis is basically cell division. It's one cell, the mother cell. Okay, one cell, the mother cell, dividing into two daughter cells. And that's the way it's usually referred. We have one mother cell, that's the original cell, and they're going to divide into the two cells, the daughter cells, which should be, if everything goes right, genetically identical to themselves. Genetically identical to themselves. They should have the exact same DNA as each other. It's the last part of the cell cycle that we went through. So we went through the G1, we went through S, we went through G2, and then I said the mitotic part. That's what we're looking at right as the mitotic part of that cell division. We're looking at this part right up here, the mitotic phase. And there are four parts of it: prophase, metaphase, anaphase, telophase, and we'll go over each of those. The phases of mitosis, we'll go over them right now, as a matter of fact. Prophase, metaphase, anaphase, and telophase. And we could put in here cytokinesis is part of that. They are not exactly the same. I tend to include them as part of this between anaphase and telophase. Actually, I consider it more of telophase, and trust me, we'll get into that. All this I should explain. We'll get into it a little bit more. Mitosis. Where did the bowling ball land? Mitosis. Okay. Anyways, going on. The way I remember that is the word ipmat. Okay. Interphase is not part of mitosis. You need to know that. Interphase is not part of mitosis, but it is part of the cell life cycle. So what is the order of the cell life cycle in all the phases? Interphase, G1, S, G2. All right. And then it goes into mitosis. P for prophase, M for metaphase, A for anaphase, and T for telophase. That's how seriously I remember the order of mitosis. Ipmat. Okay, it's not a word. It just helps me remember it. Interphase, prophase, metaphase, anaphase, telophase. All right, and you can see it here. So we go through again. We go through G1, S, G2, and then we go into prophase, metaphase, anaphase, telophase. All of this is interphase, ip-mat, all right? So what's going on in each of these phases? And this is where you probably really want to start taking good notes. Interphase, late interphase. The cell has grown. It's gone through the G2 phase. It's created the centrioles up here, the centrosome. All right, put that in a different color for you. Created the centrosome, which is the 2 centrioles, and this is in an animal cell because plant cells don't have centrioles. It's made 2 copies of the DNA, but they haven't condensed yet. They're not in chromosomes yet. They're just loose. So you have the 2 copies of the DNA. And it copies its centrosome. It makes 2 of those, all right? The pictures that you're going to see down here are, it's called a phase contrast microscope. It's where you send light in and you kind of, like a polarizing light type of thing, and therefore it mixes with the cell that you're looking at differently and produces a different light. It's just a different way of showing it. I think the most important part of these is going to be these drawings up here, these kind of drawings. They'll give you a better idea. The thing to remember about this is it's a gradual phase. It's not like it jumps from one to the other. It's kind of like we can go back to your life cycle, your lifeline. You started out as an infant, and then you go to a toddler and a child, and then a teenager, and then young adult, old fart, dead. And I'm still not there. I'm in the boomer stage, which is really just an age group, but who cares? My point, when you wake up in the morning, you don't go, oh my God, look, okay, there is one time when you wake up, oh my God, look, I'm a teenager. You went from 12 to 13, teenager. That's where we get the term. But how do you tell the difference between a toddler and a child? I don't know. It's a gradual thing. How do you tell when you're, forgetting the years, the teen years, how do you know when you become like an adult? What is an adult? Okay, it's a gradual thing. That's what puberty is. It's the development of your body into an adult body. It is not something you do suddenly. Okay, it's not like you wake up one morning, oh my God, look, I have a whole new body. Look, there's hair. I'm not going to go into that many farther. It's just assume, whoa, no, it's a gradual thing. You don't wake up one morning and go, oh my God, guys, I got to shave. What the hell? Yesterday you had a baby face, and now you've got forget the name of a beard. I knew a beard. Shut up. I knew what a beard was. My point is that you don't do, this is gradual. It goes through these phases and changes gradually. But we can identify each phase by certain landmarks and certain things that occur during the phase. So early prophase. And I would probably pause the video right now, and I would be copying down all of this stuff over here. The DNA starts to condense, the mitotic spindle, the nucleus disappears. I would probably do that if I were you. That would just be my thing. But what happens in prophase? Prophase, forget early prophase, late prophase, whatever. The DNA starts to condense. It starts to become chromosomes. The mitotic spindle apparatus begins to form. And you can see that right up here. Okay, the mitotic symbol starts to form. The chromosomes start to form. Okay? And the nucleolus starts to disappear. The nucleolus, remember, is the little ball inside the nucleus that makes ribosomes. During late prophase, the nuclear envelope breaks away. So what basically happens in prophase, all right, to sum up prophase, is the DNA condenses. I write this down. The DNA condenses. The spindle fibers start to form. The nucleus disappears. Okay? Why does the nucleus disappear? We need all the room in the cell to split up the DNA. All the other organelles have actually gone away also, so what we're end up with is just basically a cell with the DNA in it. What you'll have to remember when you look at these diagrams, this is the entire cell. That is not the nucleus, that's the entire cell. If we look back, you can see the nucleus here, all right, with the DNA in it, and then when we go into prophase, late prophase, the nucleus has already disappeared. So again, what happens in prophase? DNA condenses into chromosomes, the nucleus disappears, the spindle fibers start to form. Prophase, all right? And as it says, Pro means beginning. All right. Then we go to metaphase. Well, meta means middle. Meta means middle. All right. So basically what we're talking about is the spindle coming from the centriosomes, centrioles, the spindle fibers, all right, attach themselves to the chromosomes. They attach themselves to the centromere. So you have a spindle fiber that's coming down, and one of them attaches to that centromere, the centrosome. The other one attaches to this one. All right, and you can see that kind of happening here. This one's going that way. And the reason they're doing that is that's how they're going to move the chromosomes around. Meta means middle. Metaphase is where the chromosomes line up on the middle of the cell. They line up on the equator of the cell. It takes us to the next phase, which is anaphase. Ana means against. I don't really understand this myself. I just remember that, and I'm real serious, this looks like an A, and this, when you look at one of these, it kind of looks like an A being pulled. It's the chromosome without the A there, the A being pulled apart, okay? As dumb as that sounds, that's the way I remember it. Basically, what we have done is we have taken that chromosome and we have used the spindle fibers to pull the two parts apart. So this becomes something like this. And when we pull on the middle part, the ends trail, so they kind of split apart. That used to be an X, and now it's being pulled in to each end by the spindle fibers. Spindle fibers are just protein fibers that microtubules, like it says right up here, microtubules that can be pulled apart and be moved, all right? But they pull apart the chromosome into the two sister chromatids. Remember those, the sister chromatids, all right? We pull them apart. And that takes us to telophase. Telo means distance, all right? And basically, we have divided up the DNA. This is where cytokinesis begins to happen, and we'll talk about that in a minute. But you can start to see, the chromosomes start to decondense. They start unraveling because we need to get into the DNA. The spindle fiber disappears. The nuclear membrane starts to reform. We start forming that nucleus again. And the nucleolus begins to appear again to make the ribosomes. All right? So telophase is the end of mitosis. We've split up the DNA, and it's going to become, the nucleus is going to reform. The spindle fibers disappear, and the chromosomes start to unravel. The DNA starts to unravel. All right? And then we talk about cytokinesis. Cytokinesis is not part of mitosis, but it really is. It's splitting of the cell. Cytocell kinesis to move. We're splitting the cell. And it happens two different ways, because plant cells and animal cells are two different types of cells. So in animal cells, we form this thing called a cleavage furrow. It would literally be like if you took a balloon and you just squeezed it together. I'm going to show you a picture. In a plant cell, we lay down a cell plate. The problem with a plant cell is it has a cell wall, and we can't squeeze it in, so we have to build a cell wall between the two. Cytokinesis takes place kind of at the same time as telophase. Cleavage furrow. I love this image. A cleavage furrow is where you start, and it really starts on one part of the cell, and it starts splitting in half. You have DNA up here, and you have DNA down here, the sister chromatids, and it just starts splitting in half there, okay? The problem is every time I see this image, when we go back here, I don't see cleavage furrow. I see an old man without his dentures in. Okay? It's my bad. That's what I see. But that's a cleavage furrow, okay? Cleavage furrow comes, and it will work its way all the way around that cell until it divides in half. On the other hand, a plant cell doesn't do that. A plant cell is laying down the cell wall, cellulose. Remember cellulose? It's a carbohydrate made of a lot of different types of, not kinds, but a lot of glucose molecules strung together. And it just keeps laying down that cell wall because we can't bend we can't bend these cell walls, can't bend them in. All right, these cell walls are kind of stuck this way, so it just keeps laying down until it makes a whole new cell. All right. In plant cells, if we look at mitosis, we can look at the different phases. You have, in this case, we have interphase, ip-mat. Remember ip-mat? Okay, interphase. The DNA has been replicated, the cell's there, and you can see in the picture down here, the nucleus and nuclear material that's been stained. That's why it's red. There's a stain that combines with DNA to make it more visible to the microscope. And it's all spread out. Then we get into prophase, all right, and you can start to see the chromosomes down here. You can start to see the chromosomes thicken up. And up on the diagram, up on the top, you start seeing the nuclear membrane disappear up here. The nuclear membrane, let's try another color. The nuclear membrane disappears. You start to see the spindle fibers, all right? So you can see that better. The spindle fibers. And then we get into metaphase. The chromosomes have lined up in the middle. And in a microscope, you can see it up here. The chromosomes have lined up in the middle up here. All right, that's the diagram. Down the bottom, they don't line up that neatly. That's not really the way they work. If you see something in a microscope that looks something like this, my kids have always in the past, it looks like a spider, then it's probably metaphase. All right. And then we get to anaphase, where they split up, the splitting up of the chromosomes. And you can see again up on the diagram, them being pulled apart. Each one of these is I'm just trying to find a color I like up here. There we go. Being pulled apart towards the centrosomes, to the centrioles, by the spindle fibers. The sister chromatids are separated. And you can see that down on the bottom down here under the microscope. These are actual pictures from a microscope where you can see the chromosomes being pulled apart. And then finally, telophase, which includes cytokinesis. You need to get that used to that right now. Telophase includes cytokinesis. In the diagram, you start to see the cell. You can start seeing the cell wall being laid down, the cell plate being laid down to split it up. You start seeing the nuclear envelope come back. It starts to become two cells. And in the picture, you see the same thing. The DNA is decondensing or uncondensing, unraveling from the chromosomes, and that's why you see a big blob of that, and then you start seeing the cell plate. In animal cells, they look more like this. All right, so the question would be, this is a series, this is probably from like a fish egg or something, because if you're looking for mitosis, you look for where something's growing, eggs. And first thing we want to look for is interphase. So we're looking for interphase on this one. Interphase is going to be one where the cells are just kind of like normal. And I would say, I'm trying to find a good, no. This is kind of interphase here. This is interphase here. These are interphase. They look like normal cells. All right. And then we can go to prophase. Prophase. We're looking for where it's thickening, the DNA is thickened up. This is prophase. And I'm looking at all these pictures, and I'm saying, okay, where's the DNA thickened up? This one here could be prophase. Okay? And etc. Okay? And then we can go to metaphase. Well, metaphase, there isn't a metaphase on this one, so I have to use this. This is metaphase, all right, lined up in the middle. There was not a good diagram. Then we look at anaphase, and we can look at that. This one's pretty simple. This one is anaphase. This one's anaphase. You could say this one is anaphase. I would call it probably more telophase, all right? And that takes us to telophase. That's this one here. So, real quick to review, let's go over this. We talked about the cell cycle. We talked about G1, S, and G2, and then we talked about the mitosis. We talked about cancer cells and the HeLa cells and what happens when things go wrong. We talked about chromosomes, the sister chromatids and the centromere parts of the chromosomes. We talked about mitosis. ITMAT, interphase, is not part of mitosis. Prophase is where the chromosomes thicken up and the nuclear envelope disappears. The spindle fibers form. Metaphase, the chromosomes line up in the middle. Anaphase, the chromosomes separate. Telophase, the nuclear envelope, they've reached the poles, the nuclear envelope starts to form, the chromosomes unthicken, cytokinesis begins. And we talked about cytokinesis. Cytokinesis is where we start dividing the cell. In a plant cell, we make cell plates, laying down cellulose because we can't squeeze it. And in animal cells, we make a cleavage furrow. I hope you took good notes. If you have any questions, make sure you ask me at Edpuzzle. I'll be glad to answer them, or in the classroom. Good luck.